think we can slowly get started. So I would like to welcome everyone to today's Theoretical and Computational Biophysics seminar. Today's guest is Professor Daniel Zuckerman from Oregon Health and Science University, who agreed to give us a seminar kindly. So before I introduce Professor Zuckerman, I would like to just remind everybody of a few important technical notes about our setup. So again, we are promoting everybody to panelists. That means you have full privilege to turn on your video, turn on your audio, microphone, and interact if you wanted to. It's really like our kind of physical TCBG seminars. We try to keep it as similar as possible. But then I would ask everybody to turn off their mics at the beginning of the seminar so we minimize background noise and interference with the seminar. You can keep your cameras on actually if your connection is good enough. So that's again, that gives a more interactive impression to the speaker and the rest of us. And then if you have any questions, please type them in the chat box. The Q&A only works with non-panelists. And in this case, we are promoting everybody to a panelist status. So therefore, you cannot use the Q&A box. Put them in the chat box, and then maybe in the middle of the seminar, if it's a very urgent question, or at the end, we're going to actually go through the questions and ask you, if you have time, to repeat your question yourself. Turn on your camera and interact with the speaker. So that's what we are trying to do. So that way, it's going to be a little bit more personal. So with that, let me actually introduce our speaker. Dan actually studied religion in the beginning at Harvard, and then he switched to physics and received his master's of degree from UCLA, and then his PhD from University of Maryland, College Park, where he actually worked on statistical physics and electrolytes. And then he did a postdoc at Johns Hopkins University with Tom Wolfe, whom we know, one of our close friends and collaborators at some point. During which he actually focused on developing methodologies for equilibrium and non-equilibrium simulations of biomolecular systems. So that was his way to coming into more biophysical areas. But he kept his focus on free energy calculation and calculating reaction paths and reaction states and rates, which are actually still a very, very fundamental, very important aspects of this business for all of us. Then he joins University of Pittsburgh in 2002 as a faculty member and goes through the ranks there as the department also evolved their Department of Computational Biology, becoming computational and systems biology. And then at the same time, he was actually initially associate director and later the director of the joint PhD program in computational biology between Pitt and Carnegie Mellon University. And then he was also vice chair of education for Department of Computational System Biology between 2014 and 2016. And in 2016, he moved to Oregon Health and Science University into Department of Biomedical Engineering, where he's a full professor since then. So he, again, I think research-wise, Dan really stayed focused on the very important aspects of molecular simulation, namely finding actually transition paths and rare events, how to sample rare events in molecular simulation. These are all very important and fundamental and basic things for us, molecular simulators. And he has been very carefully actually developing extended weighted ensemble approaches, detailed study of conformational transitions in proteins that are getting more and more complicated in his studies. Transition path time is another area he has been working on. And also actually discrete state kinetic modeling is actually a sort of recent, as far as I know, work that he has done again on very complicated systems like rotary ATPases. He has, in addition to all the contribution as a really hardcore statistical mechanician to equilibrium, non-equilibrium processes, he has always actually kept an eye on education and writing really useful articles and books. I really admire him. So I have one of his physical books in my home, and I just started actually checking your online book, Dan. The lens on the cell, the biophysical lens on the cell, which I highly recommend. I can paste actually the link to this. This is all free, available, very simplified description of very important concepts in the statistical mechanics of molecular system. I really encourage all the students, especially from non-physics area, who would like to learn more about this, to examine this online book from them. So thank you very much for all your contributions and for accepting our invitation, Dan. So we look forward to your seminar.
Oh, hey, Ahmad, thanks. That was a very, very kind introduction. I really appreciate it. And yeah, no, I've known Ahmad and of course before that his work for a long time. And yeah, so it's a special pleasure to be speaking to you know, I think one of the most important modeling groups in the country, in the world. So I hope we can learn from each other. And I'm going to just look out of that. I'm going to share my screen.
Mm-hmm. Perfect.
Okay, can everyone see the pointer?
Yes.
Yeah, okay.
Yes.
So yeah, and thanks everyone for taking time. I know it sometimes can be even trickier despite the fact that we may all be at home. And I would actually welcome you to interrupt in the middle. I think we have a number of students on the call, and so if you have a question, probably someone else does, and I'll try to pause a couple of times as well, because I want to talk about actually 3 stories, I would say in descending order of time commitment. First, I'll be talking about molecular machines, then we'll be turning to Markov state models and kind of non-Markovian variant, which we have branded as history augmented Markov models. And then Ahmad had mentioned this weighted ensemble approach, which I won't have time to discuss in much detail, except to talk about why we need, why we need these history augmented Markov models in that context, actually. As Ahmad said, these problems are still very difficult. So that's the short version. So I want to talk for the first part of this about machines. I know everyone associated with Ahmad is well familiar with them, and I'm not going to belabor anything here. I'm just showing a few special friends in this poster that came from the Protein Data Bank group. And and I'll mention one thing. The ribosome is a machine which nominally is the translator of genetic information into proteins, but of course it's also a proofreader. So it's a very cool thing, and it's something I want to return to. Now, there's really only 4 things that a machine can do. And so all of the magic that those various machines from the previous slide do actually just come from different combinations of these processes, and with this sort of invisible ingredient, which we might call coupling. It's not that everything possible can happen at any time. It's actually that many of these processes end up being coupled together, and that's what enables the cell to do what it needs to do, and using free energy to do so. And I would say that besides those 4 ingredients, you shouldn't have too many other expectations. And it's because there aren't really, besides the rules of physics and physical chemistry and evolution, it's not clear that there are any additional rules to biology. We love to have categories, give things names, and so forth. That doesn't make them true. And there's, one last point that I'll just highlight here is, that there's also no rule for simplicity in biology, unless it's somehow evolutionarily favorable. And this is just a list of some, if you will, unexpected functions that have arisen in recent years, and I'm sure you can think of many others yourself. And so we're going to sort of bring this kind of skepticism to some of the cartoons in the business, starting with this one, which is from that venerable source, Wikipedia. And here we're looking at the ribosome. And basically what we're seeing is that, tRNA molecules loaded with amino acids are matching to the mRNAs, and then the amino acid chain is getting synthesized accordingly. And that's sort of, roughly true at the high level, but as John Hopfield and Jacques Nina showed quite a few years ago now, 35 years ago, and that in work that should get a Nobel Prize still, because John Hopfield's still alive, they showed that rather than this sort of kind of textbook view of which I just described, of the tRNAs coming on and infallibly adding to the growing polypeptide chain. In fact, there's another step that seems highly counterintuitive, certainly not the simplest way to do things. In fact, the cell spends free energy in form of GTP being hydrolyzed in order to slow down this process and, in fact, create a high probability that a given tRNA will unbind. And it turns out that this is proofreading. And we're not going to have time to go into the details of it today. I do have some discussion of this on my website that Ahmad mentioned. It's really just kind of the coolest thing, and motivates some of the questions that I'm going to bring to you in a few minutes. So this is what the ribosome more or less really does. It does this extra step. It spends free energy, but it increases the accuracy of translation to the point where we're all here today instead of being in the muck. Actually, I don't even know if we'd be in the muck without this because bacteria do it too. Okay, so now I want to go to a cartoon that I'm just sketching for you here. And this is a cartoon of a symporter, and they're just showing 2 different types of molecules, but think of A as an ion, like a sodium ion. And we know there's a gradient of sodium ions across the plasma membrane. So there's a driving force from outward to inward. And think of B as some molecule that the cell wants to import. I just want to take you through this cycle as sort of a reference point for some of the future stuff we'll talk about. So in this cartoon, which again is just a cartoon, first the ion binds, that was A binding to the outward facing conformation, that's the extracellular side on top. And then the B molecule binds, inducing a conformational change. This is sort of the coupling that I was mentioning before. Then when it's facing the intracellular side, then the 2 molecules unbind in turn and the system resets. And this can go on so long as there's a net driving force. In other words, that the driving force that's pushing A from outside to inside is at least as great as the driving force opposing that. So, and in the case of the cell, the driving force from sodium can be strong enough to actually import substrates opposed to their gradients. So in other words, even if the concentration is higher inside. And this is one of the ways the cell does business, and there's lots of variations on this with different stoichiometries and so forth. But I want to just stick to this one-to-one, so one-to-one meaning 1A and 1B for a moment, and point out that what we're seeing here is the AA bind first, B bind second, and then B unbind first, A unbind second. So it's a particular order of these events. But if you think about it, these 6 states aren't actually all the states that might be possible in this kind of system. And in fact, there's a couple others, just if we're just including the conformational change, single conformational change, in other words, 2 conformational states. So in particular, in the outward facing, you never see B by itself. And in the inward facing, we never see B by itself bound. And this just reflects the, so there are 2 additional states that are possible. So those are the 8 that are being shown here. So there's 4 possible for each conformation. And then it turns out that there's actually 3 other equally valid in principle paths for this co-transport or symport, as it's sometimes called. And, you know, just depending on the order of which binds first, A or B on the outside, and then A or B unbinding on the inside. So that's interesting because it raises a lot of questions about what's really happening in some of these transporters. What's really the order of events if you want to understand the mechanism. And to make things even worse, if you think about it, you know, the path that's taken just depends on the rate constant. So what, you know, a given path here is sort of showing that, well, this rate constant is finite and that rate constant is 0. But what if they're comparable? Well, then you could have a mixture of these models, right? So there's actually an infinite number of one-to-one ideal symporters, and we're not even talking about, you know, leaks or anything complicated like that. So just in this toy sort of thought experiment, there's a lot of questions that come up. And besides just theorists scribbling on cartoons, the experimentalists have been at work, and there's a number of recent studies suggesting that some of these machines use more complicated mechanisms than were previously thought. I'm not showing references, but there are others. There's cases, well-known cases, where the stoichiometry is not an integer. So there's different stuff going on. In many of the plasma membrane transporters, it's known that water is co-transported stoichiometrically. And there's other models where you see something happening, like in this case, this is a channel that's gated by ATP. It's not driven by it, but it nevertheless still hydrolyzes in ATP. And so it's sort of a mystery. Why would that happen? And there's lots of questions, like every system just about can raise some interesting questions, and I don't want to spend a lot of time on them, but I will say that there is plenty of experimental evidence for kind of a more nuanced view of how these transporters work, rather than just the kind of cartoons that we may be used to seeing, where there's sort of, you know, arrows drawn between known structures. And I think this, you know, can be a little misleading. I mean, here I'm just listing some of the experimental methods for studying transport, and all these methods have their limitations. I'm not the first one to say so. And certainly we all know about number 3 here, where we don't have enough simulation time to do what we want. But I want to suggest that we can maybe enhance our understanding, or begin to, by kind of examining a 4th sort of approach, which is kind of a systems biology approach, which I'm going to talk about now. So again, so the systems biology approach in brief maybe is to kind of, or the ideal systems biology approach might be to consider all possibilities if you could. So could we kind of figure out a way to do that, to consider all these different possibilities? And what we're going to do is kind of steal a few ideas that have been around and that I'm sure you're familiar with. And in particular, we're going to actually do a metropolis Monte Carlo simulation, not of an atomistic model, but in the space of models, okay, by actually perturbing rate constants and seeing what we find and trying to do that in a way that leads us to different mechanisms than we might otherwise find. And this is going to get us away from assuming there's a mechanism. We're going to try to use this as a search/sampling method to find an array of mechanisms that might be consistent with a given function. So just in a little more detail, we'll start from some initial model. This is showing one of those ideal transport paths. In fact, we actually start from kind of a nonsense model with just random rate constants. In each Monte Carlo move, we'll be adjusting some rate or one or two. And actually, the way we do that, though, is we actually represent all the rate constants in an energy or free energy space. And the reason for that is that that guarantees that you satisfy cycle constraints. And there's quite a few here. And we didn't want to have a code that would go hunting through and dealing with that. But if you work in energy space, you're kind of guaranteed, you are guaranteed to preserve those cycle constraints. So we'll change either a barrier height or a state energy. And then we take the resulting rates and we'll solve for the steady state of the system. So this is we're just gonna have a set of ordinary differential equations. That's what this is sort of a shorthand for. We're gonna solve for the steady state of the particular model. And that steady state is gonna give us the flows along all the edges in the steady state. And then we're going to have some effective fitness function. So this is not a physical energy. This is something that we're just making up that's gonna help get the function that we want. In this case, it's going to be something, we're always going to start with minus the flow of the substrate. And that's going to mean lower energy when substrate is coming into the cell. And that's going to actually be against its gradient. And then we're going to go ahead, accept, reject, and then go on. And then there's going to be this essentially fictitious temperature parameter here that we get to play with a little bit, and I'll mention that in a sec. But this is the basic approach of what we're going to do. And then for people who know about these types of models, we're actually going to use effective first order rate constants because sometimes we have binding events, right? So we have to include the concentration that keeps everything on the same basis, makes this energy formulation consistent, and this is all figured out by Terrell Hill long ago. So here's an example of a trajectory that we've run. And this is actually a tempering simulation. So that effective temperature, that beta is like 1 over temperature. We're raising and lowering that to look for models. So it's a little hard to see, but on the upper left, we actually start with this kind of nonsense model. So the energy is positive. And then it will decrease, and then we're going to raise the temperature. And then you see that we tend to find a lot of models with these are all negative energy. So when there's negative energy, again, this is the Monte Carlo energy. So it's the fitness that's minus the flow. And so all these are working models. All of these models use the free energy of the sodium gradient to pump in a substrate against its gradient. Okay, so we're finding all these different models here. And so when we do this, we first wanted to do kind of a reality check and, do indeed find these idealized one-to-one symport models when we set it up in that way. So we really set the kind of the conditions under which we're running the simulation because we say what the concentrations are in and out of the different molecules. So when we set them up for symport, we find symport, okay, that's good. And then if you measure the currents, then you see that it's 1-to-1. And of course, we didn't allow any possibility of anything weird like a leak, so that's no surprise. Perhaps somewhat less expected, although I gave this one away already, is that you can actually have different types, you can have mixtures essentially of those different ideal mechanisms. And I'll just for reference, so our convention here is that we've taken all the flows. So these are the net steady state fluxes, and we've normalized them to the maximum just to make it easy to look at. So there's one edge that's always going to have one, and everything else is kind of going to be a fraction of that. But this is also ideal one-to-one symport. It's using all the 4 paths, and you just see, again, the currents are one-to-one, just as you expect. And in these graphs, by the way, I'm showing this dashed vertical line that actually is the condition under which we ran the simulation. Just for reference, you'll see that's going to come up in a moment.
So this is very interesting, Dan, so if I may interrupt you. So in the purple case, in terms of the order of binding of blue and green, you have almost half and half. In the blue case, it's like 30%, 70% kind of. It seems that one of the substrates, one of the transport species, somehow in this model is preferred to be released or bound in that inward facing conformation. Do I read this correctly? From the
okay. In this particular model, yes, right, the rate constants are favoring a certain path, right, over one path over the other. Now, so don't forget, where our whole thing, our whole sort of sampling thing is a stochastic process, and it's not, neither the energy nor the temperature are real. So bear that in mind. So we're just finding, if you will, candidate models. So you shouldn't interpret it as this is saying like, oh, based on these conditions, it was 70-30, because, under the same conditions, we found all these other models. So at this point, and I want to get back to some issues that are implicit here, we're just kind of, if you will, making a list of everything that's possible. That's what we're trying to do.
Okay. Mm-hmm.
Okay. Any other questions?
No.
So, and you can probably imagine that if we changed up the concentrations, we could use the same method to find anti-porters and so forth. And sure, we can do that. But then we want to bring up a different possibility that kind of brings together a couple of the themes I mentioned. And this has to do with a sugar transporter that collaborators of mine have studied for a long time, namely Michael Grabe and John Rosenberg. And they work there, who are also, well, for the purposes of this talk, they're also modelers and theorists. And they work with some experimental groups that study the system in the lab. But this is an organism, or this particular transport, excuse me, comes from the Vibrio organism. That's the V in the VSGLT. And Vibrio kind of lives in the muck. And unlike our own cells, might have a kind of higher motivation for not just bringing in the good molecules, but keeping out the bad molecules. And so the question that John and Michael ran some molecular dynamics simulations. They saw some unexpected behavior, and I don't know if this was just stochasticity or there was something real to it, but what they saw was they would see ions permeating through without being coupled to transport, and they would also see more frequent unbinding of the sugar back out to the extracellular region in their simulations. Okay, so we know that simulations are an imperfect tool, but it was suggestive. And John raised the question about whether a transporter can proofread. And that's the question that they were thinking about it, and sort of brought me into the conversation. And it turned out to be just a pretty interesting theoretical exercise, which I want to tell you about now. So if we're going to allow, so the color code, again, the blue is the ion, the green is the substrate that we want. And if we want to talk about discrimination for a kind of false substrate, which I'm going to call a decoy, and sometimes labeled W for wrong, that's kind of evoking the Hopfield's notation, then you you have these two additional states that come in, two additional binding states that come in for each conformation. So it gets to be more complicated, and you could, you could try to just, draw lines through this space and study them and see how that works. But we decided we wanted to see if we could really sample the models that are consistent with not only importing the correct substrate, but suppressing the wrong one. So to do that, we're going to use our same Monte Carlo approach, but now we're going to modify our fitness function so that there's a ratio of the true substrate over this decoy. We just wanted to see if this would work. Oh yeah, one other technical detail here that's important. So if I want to get enhanced selectivity for the true substrate over the wrong one, there's one very easy way to do it, right? I could just take any of the rates for the W processes and just make the barriers really high, right? That's going to work for sure. But that's not what we're talking about. So we want to build into the model that this is really a decoy. This is a molecule that's similar to the true substrate, but maybe it has some different moieties on it. And so the way we're going to do that is we're going to let the system know that that one thing about this decoy is that it binds more weakly. And in fact, that's the only thing that we're going to change. So this is basically going to change some off rates. We're going to assume 1 kT weaker binding, but all the on rates are going to stay the same. So basically, we're not going to allow, if you change a barrier height for the true substrate, you have to change it for the wrong one too. So those things are actually tied together under the hood. And speaking of under the hood, that was actually most of the work, needless to say, to put this together. And that was to build in all the different you need mutually exclusive binding. As I mentioned, you needed certain rate constants to be tied to one another. So all that stuff was a lot of software engineering that we dealt with. And primarily it was this student, August, who did this work. So let me show you what happens. We do the same, thing. I'm afraid I'm showing you the exact same panel. I don't know which system it was for, but it'll look like this because again, we were doing the tempering. And then, as I mentioned, all these models with a negative energy are valid models, right? Not just the ones at the minima, but all of them, anything. So, and we don't, this fit, just like in MD simulation, if I told you the energy of a conformation and asked you based on the energy to tell me what it was about, you wouldn't know. Likewise here, we don't know what the model means just by the energy. So we want to really examine all of these. And so this is where we took another sort of idea from systems biology and we did clustering. We just basically set up a distance based on these flows that I mentioned before, these normalized flows. So you can just make them all into a list like a vector, and you can think of a distance, based on a dot product, or we actually just did a Cartesian distance for these guys. And then, we cluster them into these different groups. Hierarchically, you can cut it anywhere, but I'm just going to show you based at the level of 4 different models. And here's one of them. And this one I've chosen to highlight because it's a little bit easier to understand, not from this picture, but hopefully the pictures I show you in a sec. But again, it's just like all the flows are normalized to the maximum. And you see there's a lot of different states involved. So let's see if we can kind of just dissect this out by basically picking out the mechanisms that we can understand intuitively. And there's no unique way to do this, but there's different ways. This is an informative one. So on the lower left here, what you're seeing are actually the ideal symport pathways. So this would be like 1-to-1 symport. Okay, and now here's some little variation in which order, but basically it's 1-to-1 symport. These other pathways, which have this kind of mirror symmetry, you might notice, those are just leaks. Okay, so let me explain what I mean by it when I say it's a leak. Let's just follow the pathway through. So you have the outward facing unbound, substrate binds, the ion binds, the substrate unbinds, there's a conformational transition and the ion unbinds, and then it goes back. So the net result is just that each time you do this, one blue sodium goes down its gradient and nothing else happens. And this this pathway occurs on both sides of the system. In other words, including the true substrate and the decoy. Excuse me. So when I come back to these flow diagrams and go through it, remember plotting here on the y-axis the flow. The x-axis is the chemical potential difference of the driving sodium ions. And as I mentioned, the dashed line is where we actually ran the simulation. So we're running, you could set this up in different ways, but we were just using one condition here. And so at this condition, the blue is the sodium flow. So that's the that's the biggest flow, right? And then you can see that, some fraction of that flow, somewhat less than less than half is the substrate. But look at the decoy flow. It's actually exactly 0 at this particular unique single point, right? So in other words, this metropolis really did what we asked it to do, which is nice. Always be careful what you ask for. But in this case, we said, go find, optimize that fitness. And it found a set of rate constants such that there's actually 0 flow. And in fact, there's actually a little regime where you're actually pumping out the decoy, even when the substrate is coming in. So in other words, the green is positive and the red is negative in this little regime. So this is strange, but this is what you get from this. So when I said at the beginning that who knows what biology is doing and who knows what biology is optimizing and who says it has to be simple, this is the kind of thing that you may get out of. And I want to show you that this is actually the thing that causes the discrimination is actually the leak, and that would be very much an analogy to Hopfield's model of proofreading. And the way we tested that was we simply, if you go back, sorry, let me go back to here. You see all the both leak pathways go through this central transition. So we basically just cut that one off. We went into the model, set that barrier height to a large number. And then, so there's no flow through the middle. What you see is really just this kind of ideal symport pathways, but for both substrate and decoy. And then you get, there's an expected ratio that you get based on that 1 KT binding difference. And so that's what you see throughout. And there's no, the only point at which the decoy flow goes to 0 is when all the flows go to 0, right? So this is like a balance point. It's a kind of constrained equilibrium. So that's just one model. Here's some others that emerge from the cluster, and we won't go through them because they're similar, but they're hard to understand. And I would say that we could never really guess these models, let alone the spectrum of them. And so just to repeat what I was saying, just beware of what you assume when you're doing these models. What we wanted to do here was really present a theoretical picture of how to think about heterogeneity, how to look for it. And I wish I could claim that everything was perfectly original. I tried to give credit, but this from 1977, we see this Barry Honig article. When one tries to choose between models, it'll be almost certain that the choice is to some extent an arbitrary one, and that most systems will be it's a mixture of different models. Yeah, so anyway, smart people thought of these things just a few years earlier. So we're following in their footsteps. And we see this really as more of the beginning of the process. So, that was a totally theoretical exercise, really asking the question, could it transport or proofread using the ion gradient? And that, we showed that it could. I think in the future, we need to really work more closely with experiments to see, and and we can find the set of models that are consistent with the data and hopefully propose additional experiments. And I think a big open question here, one that we're just starting to look at, and this is in collaboration with the Hensler-Wildman group, is really what can you expect in the best case scenario? You know, how, let's say you had all the experimental data you wanted, would you still be able to figure out, would you be able to figure out the mechanism, or how much would you be able to narrow down the space of possibilities? So that's a question that we're looking at right now. And so stay tuned. So any questions then on, I think that ends, yeah, that ends this section if there's any further questions.
So I see there are a couple of questions in the chat box. Dimon, you want to ask your question? Dimon Ray?
Yeah. So my question was like, when you make the Monte Carlo moves in the model space, do you really need the experimental rate constants beforehand to make these models? And as a follow-up, like, is it possible to predict rates between certain states in the mechanism when the experimental data is not available?
So no, we don't need the experimental rate. The whole modeling pipeline, you don't need the experimental rates to do that. It's just a question if you want to find models consistent with the data. Now you could put in some measured rate constants, but I think there's also going to be ambiguity about what exactly was measured, right? You may not know, you know, which exact edge to put that rate on. It may be obvious in some cases. So there's a lot of things that we can play with there. And yeah, so I think that the goal is to go into a system, you know, with a lot of the microscopic information missing, and saying how much of that can we infer by doing, let's say, a set of experiments. So that's sort of the bigger picture.
Shashank?
Yeah. So I have a question like similar to Dhiman, which is more on the constraints part. So the question is like, can we alter the constraints or the restraints that you are applying in the system in your Monte Carlo model, let's say on substrate, to get some insights on the evolutionary dynamics or like evolutionary molecular biology kind of thing, like how transporters or a particular system under study evolve?
Yeah, so that's a great question. And I've sort of wondered about that. I think, you know, because I think what you're alluding to is that the Metropolis Monte Carlo is itself a kind of like evolution in model space. And so the only question is then which of our, you know, abstract theoretical trial moves are actually physically realizable. And so that could be a fascinating thing to try to study, like in other words, which rate constants can you individually change by mutations and so forth. But yeah, so that's a whole long discussion, but good, yeah, great question.
Right. Yeah.
Yeah. Hi, Doctor. In your schematics describing that, I think it was a few slides ago with your equation. Before that, yeah, I think it's good. Yeah. So here, so when you're calculating those rates, or I think do you call them fluxes? Is it like and when you apply the steady state assumption, so it's basically like you're calculating fluxes between one state to the other, right?
Yeah. So when you solve the steady, so it's when you solve the steady state, you actually, you know, you solve directly for the populations of the different states, and then you know the flows based on the product of the populations and the rate constants, and then the net flows, of course, come from taking the difference between the two directions.
Oh, so it is actually bidirectional, and you
yeah.
Okay, so if that is the case, like I think in your some of the illustrations, like some of the states are not connected, probably because maybe it's not a valid transition. Like, you know, when there's no ions and when there's a blue ion, you don't really connect the flux there.
I see. Yeah. So let me just address that a little bit. So right. So first of all, these are the net fluxes that are being shown. So there's always flux in both directions. So when you see nothing here, and I want to remind you that the barrier heights are the same on both sides. So these are non-zero rate constants. What it means is that there's a balance, that the flow is balanced in the two directions.
Oh, okay. But
so yeah, you can't, yeah, there's a lot more information that I'm sort of withholding. And again, this is just the one particular, this is just the point where we did the simulation. If you move to the left or the right, if you change that driving force a little bit on the ion, then you'll pick up net flows.
yeah,
Those edges are still active. Yeah.
Thank you so much. I was a bit confused. So when you first initialize your simulation, like at the very beginning of your Monte Carlo simulation, do you not have to have certain type of like equilibrium concentration for each state? Don't you have to have that kind of thing?
Well, so maybe you're we have to
initial condition.
The initial condition. So remember when I said that all the rate constants are formulated in terms of energies, and that's to keep thermodynamic consistency. So that's the only necessary condition. Don't forget, none of the conditions, sorry, here are equilibrium. This is in a condition of driving. So you may be thinking of the principal detail balance. So we have to make sure that our system would obey detail balance when it goes to equilibrium, but we're actually simulating it out of equilibrium. It's a little bit, there's some subtleties there, but that's the short.
Oh, okay. So detailed balance as in the within the context of formulating Markov states. Is that what you mean?
Right.
Okay.
And that's built into this thermodynamic cycles that I've been mentioning.
All right. Thank you so much.
Okay, thanks,
all right, Shell, I can't see the chat, but I...
Good. You have addressed all the questions very nicely. Please continue.
Yeah, okay, thanks. So let's go on just so you get a taste of the second part before we run out of time. So I was going to talk quickly about Markov models, which I think are having, as you know, having a big impact in our field. They're widely used, and they're often used to discuss mechanism. And I think that's something that we need to think carefully about. So just briefly, again, I'm sure you're familiar, but a Markov model, you take this trajectory, which is this dotted line, and you discretize your space, right? And then you can calculate a transition matrix of conditional transition probabilities amongst these states. And when you do that, you can calculate basically anything you want. And of course, you should be careful and sort of validate your model. That's what's being shown here, is that you should show that it exhibits a kind of Markovian behavior, which corresponds in part to a plateau in these implied timescales. As I said, it's when you're building a Markov model, you're just doing the conditional transition probabilities between these pairs of states, just based on 2 steps in time. So you're not using history information that's available in trajectories. And that's why we came up, or that's actually not why, but we came up with this other formulation actually to help us with weighted ensemble. But it turns out to basically be a good way to generalize a Markov model. So if you think about it, if you're looking at a conformational transition process, right, and imagine hypothetically we had this very long trajectory, we can color it in terms of whether the trajectory was most recently in A or most recently in B. So that's the red and blue. And then in this formulation, you can actually build, if you will, 2 Markov models. Neither of them is really Markovian anymore, but if you build a Markov model based only on the red trajectories, and of course they don't go straight across like this, they're stochastic, right? And also from the blue, you would have 2 different transition matrices, okay? Neither would be a true Markov model, but they look like a Markov model. And so that's sort of what's just being shown schematically. It's almost like a 2x2. And I'm going to use the notation that alpha is this A to B direction and beta is the B to A. And that's why you have these. So in addition to just having the rate constants, or more for technically the transition probabilities, you have them conditional on whether you use the red trajectories or the blue. And so and there's a direct relation between these two formulations. So it starts from thinking about it as a single discrete region, which they will call a microstate in the Markov community. And the equilibrium probability of the microstate is just the sum of the two, the alpha and the beta contributions. Here I'm showing you not a single long trajectory, but like an ensemble of trajectories, each from those separate directional sets. And then likewise, the transition probability is of a Markov model, or the one that's kind of corresponds to equilibrium, is just a weighted sum of the transition probabilities that you would get in the two ensembles. So there's a direct connection between these two things. But the nice thing about separating out this way is you get to a kind of magical property. And what I'm showing you here is actually what we call the discrete hill relation, and it's the same hill, although he didn't work on this trajectory part, but it's actually still based on some of his ideas. So in particular, the inverse first passage time is exactly equal to the steady state flow, if you will, from all the microstates not in B to all the microstates in B. In other words, it's the net flux into state B, okay? And the key thing here is these superscripts. You see those red superscripts? If I take those away, this is no longer correct, okay? This is not generally true in a Markov model, but in this what we call a history augmented Markov model. So history in the sense that you remember where you came from, which direction. This is exactly true regardless of how big or small the states are. They're not assumed to be Markovian, and at any lag time that you care to use. So, and the lag time is just the time difference between, like the time between, if you will, frames that you use in your analysis. So this is a very powerful bit of information, and it's saying if somehow we could get these labels, we could, stop worrying about Markov assumptions and stuff like that. So what I'm going to show you is that how this analysis performs by comparison to regular Markov models by looking at some of these very long trajectories that were, folding and unfolding trajectories that were run by the David Shaw group. And we're going to examine a few different systems. So first, the models were built by people who know how to build them, namely from Frank Noé and John Kodera's group. They were validated using the usual tests. And here you're seeing these implied timescale plots. And what's different about these, there's a couple of things that are different from what you're probably used to seeing. First of all, they're on a linear scale. And the reason is that anything that's on a log scale looks like it's plateauing, even when it's not. And if you don't believe me, plot a straight line on the log scale. And but the other thing about these is notice the lag times when these do level off. They tend to be in the 100 to 200 nanosecond range. So don't forget, that's how long we were running simulations just a couple of weeks ago. But that's, in other words, that's the lag time that has to be below that. It's not a valid model, right? You see what goes wrong. Right, these timescales are not level. So I wanted just to show you the results. Let me just do one quick technical thing, and then I'll show you a couple of quick results, and we'll try to wrap up. But basically, if you do a close up on one of these trajectories that entered state A, and then it hung around there for a while until it made a transition. we can define a couple of timescales from this. First of all, the first passage time is the total length of this whole process. And second, the event duration or the transition path time is just this dashed line from the last exit from A to the first entry to B. And one other technical issue to point out is that you can look at this trajectory, I'm trying to suggest with these dots, at different time discretizations. So you can almost think of that as like a lag time or discretization time. And I'm talking about analyzing the MD simulation, okay? So this is not a model, this is just a kind of an examination. And so anyway, the first passage time is gonna change based on the time discretization because you may miss the first entry. So it actually increases monotonically with longer time discretization or lag time. So all that is background so you can understand this graph, which is complicated. So what's being plotted here on the x-axis is the lag time. So for a Markov model, it means what you think it means, the lag time they use in their analysis. But for the MD, it means the discretization time I was just mentioning. And so the green bar is the exact result from those long MD simulations. And it has this characteristic shape whereby, let me just go backwards, at very long lag times, these lag times are so long, they're basically as long as the first passage time. So this is actually kind of nonsense. This is just, this is not kinetic behavior. This is taking off into trivial behavior, basically landing in one state or the other at random. But this sort of plateau thing is kind of a nice feature. It makes you think in physical terms, maybe the first passage time isn't that sensitive to the discretization, right? If we think of the green. And then if you will, there's the mathematician's first passage time, which would be literally the first entry point, would be kind of off to the left somewhere. We'd have to see it plateaus the other way to see that. But what you see, again, is typical for all these results, the different proteins in the different directions, is that the Markov model does not yield the correct first passage time until you get to quite long lag times. But it's also true that the same lag times that I mentioned before are the right ones for getting into this sort of sweet spot. Now, they don't all have a nice plateau, and there's no rule that they have to have a plateau. This is based on this choice of states. I haven't described that to you. There's lots of issues that come in here. But anyway, this is the typical behavior. The blue lines are different kinds of those haMSMs, and the solid blue is kind of the exact one that is exact. So we knew it was going to match the MD. But the other ones are saying, what if we just use a little bit of history, just a few nanoseconds? Could we still do better than the Markov model? So the answer is yes, that you can thread through this result here. Now, I see that I am out of time. I'm just going to mention to you that when we also, so I'm going to mention two things to you. First is that the transition path time in these, if you go to the MD simulations, is extremely short, much shorter than the lag time. And that tells you that you shouldn't use the Markov model to analyze mechanism in these systems. You just should not do it. You probably should be aware of doing it in any system. This is one where we happen to have enough data to really know what's going on. Nevertheless, if you did choose to do it, what would happen? Would it turn out to just be okay? Would it? And what don't have time to go into is these different graphs. And unfortunately, this is from a paper which not only is unpublished, it hasn't. It's not a preprint yet. It should be within a couple of weeks. We are negotiating the final political details of this study. But the bottom line is the Markov models do not match any kind of mechanistic observables in any kind of consistent ways, particularly not at the longer lag times. And so again, the blue is kind of matching the green and the red is all over the map. And I don't have time to go into those different analyses. But I will also mention before ending that we were able to use the same formulation of the haMSMs to help us analyze weighted ensemble data. And because weighted ensemble data as beautiful as the idea is, has a lot of variance in it. And so we could use actually these analyses to help us get to the correct answer from transient data. And so you can read about that a little bit in our Jack's paper from last year, and then there's a new paper. So let me thank the folks who did the work. August basically did all the first part, and Ernesto got the haMSM ball rolling. And this is Rafael from John Kadera's group, and Jeremy did the application to Ensemble. Okay, so sorry to.
No. Thank you very much, Dan. So we can't clap. I guess we can, but you wouldn't hear most of it. Thank you very much for a very interesting talk. I knew about your kinetic model, but it was very nice to hear again about how powerful that could be actually, really modeling. I had a question there, and again, then we can open it to other attendees. So, like, I mean, we can imagine many intermediates and then many connections between them, you know. So, but I mean, where do you draw the line between? I know that you are trying to predict the phenotype. In the example you showed us, what happens if we have a leak, or what happens if you optimize it not to go after the decoy transport and things like that. So you are imposing a kind of phenotype, and then you see based on the number of intermediates you have, what would be the optimal connectivity between these intermediates that would bring that to you. But where do you draw the line between, let's say, noise? I mean, these things fluctuate back and forth. Many other things can also happen. It can be added to the model. Is it sensitive to have too many intermediates, your model, in order to predict the optimal configuration for a particular phenotype?
Yeah. So okay, great question. So just to put it in slightly different words. You could imagine, so many of us are familiar with atomistic simulations, so we know that the real systems have many degrees of freedom, right? And you know, you could think of one of these Markov-like models with many hundreds of states, and I'm showing you models with 6 states or 8 states, right? And so there, if you will, there's always going to be a hierarchy of descriptions. I think what the burden on us is going to be to sort of understand, as you mentioned, based on the noise that's in the data, what level of description we can hope to provide. So it could be the case that, for example, you know, well, we know that this particular transporter is powered by 2 protons, but we just don't have the resolving power maybe to figure out the order between everything. And we may have to limit ourselves to sort of classes of models. And that's why I say, like, we're very much at the beginning of this. And like, the question, what can you figure out with the kinds of data? And one of the things that we're hoping will be helpful is to do experiments under a lot of conditions. And I know that, you know, traditionally the electrophysiology experiments can be quite difficult. They still can do multiple conditions, but some of the new technologies are enabling them to do things faster and study a wider range of conditions. And we're hoping that additional information will have a payoff in terms of, but the honest truth is that we don't know how good we're going to get. Sorry.
This is very interesting. You are using the term systems biology for one particular protein at work. This is not a traditional way, but this is indeed, you have a systems that is in work, and that can give you actually different outcomes. I really like it. I think it's very powerful, and then with more experimental data and conditions that we can impose on the model, we can learn a lot actually. This is really exciting.
Thanks. I hope so. Yeah, I think that in the real systems biology, their systems are, I mean, they're so underdetermined. They are like, we have a chance. I don't know if we'll get there. Okay,
so this is, the session is open. If you have questions, I know that we are a little bit over time, but we don't have any constraints here. If anybody has other questions, we asked quite a few questions during the talk. If there are other questions, you can actually turn on your microphone and ask right now, or raise your hand, or let me know.
Can I?
Yes. Seper.
Oh, I think Nandan was going to ask first, so I can.
Go ahead,
okay.
Seper. Why don't you go ahead and do your question first?
Sure. So actually, maybe I'm wrong, I don't know, but I was thinking that, you know, when you had history to your analysis, you're not having a Markovian, you know, chain model. So, you know, the process is not Markovian anymore. And when you don't have this Markovian chain, you don't satisfy the detail balance. And do you think, I know that your result matches with the long MD simulation, but do you think theoretically, does that cause any problem? Because I mean, when the process is not Markovian, the detail balance is not satisfied.
Right. So yeah, that's a good question. And so the way you can, sometimes when I talk just about that subject, we'd have more time to see, there's always the microscopic detail balance, which is like, if you think about from the molecular dynamics simulation. So that's the data that's there. And then we're doing an analysis, right? And we want it to be correct. And you can actually, you know, let me just say, maybe the shortest answer is, so long as we are adhering to the physics guiding the trajectories, governing the trajectories, we're going to be correct. And that's essentially how we derive all our results, is we sort of, you can almost do little thought experiments. So like for the two directional ensembles, right? If you have if you think about them, like running them separately, one from A and one from B, right? Like when one of those trajectories reaches the B side, right, it's going to change its color, but the net system is equilibrium. And so we know as many that change from red to blue are going to be changing from blue to red. And so that you do maintain the overall global balance. You're just doing this dissection of the equilibrium ensemble. So let me stop there, but we do worry about it. And thanks for asking.
Nandan?
Yeah, hello, Professor Zuckerman. So can I ask, the question related to is the difference between MSM and haMSM. So do we have any practical example of which method would be better than the other in kind of practical biological system?
Well, okay, you're asking a biased person, but what the data I showed you show that basically anything you can get from the MSM, you can get at least as well or better using the history information, which is always there. Now, it doesn't mean that it's sufficient. You know, that's going to depend on the data that you feed into it and how complex your system is, those types of things. But I think it means you have a better chance. You don't have to worry about the lag time. You don't have to worry about making your states quite as small. You still want small states though. So yeah, I mean, that's what all our analysis suggests.
Okay. Good. Okay. Thank you, Dan, again. So I would like to thank Dr. Zuckerman one more time for this stimulating talk.